adaptation and has multiple tasks and optimality. So all this, it's like a crystallization or a collection together, a bringing together, a tying together of the strings that we offer, right? A moment of great joy, fulfillment, satisfaction, of integration, of bringing together that which is scattered. So, the question I'm going to ask is, given circuit different system that fills out its parameter space. So just like in physics, we're going to concentrate on the simplest hydrogen atom, simplest circuit that we can solve, understand. And we're going to deal with the circuit. I'll tell you about it in a second. But just like any circuit, it has numbers on the arrows, it has parameters, right? Like all the circuits we looked at has production rates, degradation rates, binding constants. So we're going to focus on that. We saw that because of network motif, there's just a limited number of topologies of ways to connect arrows. But now we're going to look at the numbers on the arrows, the parameters. Okay, so I'm going to fix one circuit topology and ask about the parameters. Right, so this is topology, right? It's a connection pattern. Or network motif. And it has these parameters that basically determine its dynamics. And so we can think of a parameter space. And each system each instance of the circuit is a point in parameter space, right? So different systems that have the same circuit pattern, one might have this value of parameters, another one might have this value of parameters. But maybe a different same system in a different organism, different parameters. So this parameter space, it's just a kind of trait space where the traits are now the numbers of the biochemical parameters of this particular circuit. Hi, nice deep sigh of relief in your honor. And parameter space can be full, you might say. Maybe if we start looking at the same circuit, different systems, we get a cloud like this. Of course, I drew here just 2 parameters, but a circuit might have 20 parameters. So you might have a cloud like that. We collect more and more systems. Maybe, like we discussed last time, or maybe all systems fall on a curve or a triangle or something simpler. Or maybe if you look at different systems, there's only one set parameters. Maybe all systems not only have the same arrows, but have the same parameters. Okay. And why is this important? It's important, of course, because anyone here who's done experiments in biology knows it's very difficult to measure those parameters. So if we have a situation like this, if I can measure one parameter, you can deduce all the others, right? If we're talking about a one-dimensional curve. So some parameters are easy to measure, some are very difficult. So if you have a situation like that, it makes life much simpler. It says not only is there a small number of circuits, topologies, but within each circuit topology, there'll be actually a very limited range of possible parameters. So if this is the case, then not only we have a small set of motifs, each motif has a limited range of parameters. Now, right now, where we are in systems biology, we don't know which one is what's right, or I'm not aware of a situation where we have enough data on enough circuits to be able to distinguish between these. I think it's a problem of central importance if we're going to understand biological systems, just like engineers understand a device, let's say. And that's why I want to devote part of our class to thinking about this problem in a situation where we can analyze, and where there's also some data. Okay, so we're going to ask which of these situations applies, and we're going to use what we've learned to tackle this question. Any questions? Yeah, thank you.
Each space defines different parameters, but the architecture of the circuit is also, as far as I understand, it's not defined by the space, right?
Right, so maybe we should clarify that a parameter space for a given circuit topology. Each circuit topology will have its own parameter space. So we're going to consider one topology and dig into its parameters. Other questions? I'm going to erase now. So we're going to deal. The circuit we're going to choose is what's called homeostasis circuits, which basically what they do is they respond to stress or changes and bring the organism back to, I'm going to give an example, a specific example now. The homeostasis, this word is a very common word, we use the word in physiology, and a lot of things that organisms do, according to what we think, is try to bring themselves back to keep a situation where you can be staying alive. So, for example, the heat shock system. E. coli. E. coli can survive different temperatures. It can survive low temperatures like 10 degrees. It really grows fastest at 37 degrees. If you go above 37 degrees, it starts growing slower and slower. 42 degrees, it dies. If you give a brief pulse of 42 degrees, yeah, it can survive. And the way it's, the problem with heat shock, or one of the way heat shock kills organisms, has to, and E. coli too, is so temperature. We're going to call this U here. This makes proteins unfold. And Y will be the concentration of unfolded proteins. Hi. You know, you also deserve a nice deep sigh of relief. Why not? So this is the damage. Why are unfolded proteins not good for the cell? They have this hydrophobic parts sticking out, and they tend to make aggregates and stick to things and clump things up. And the cell basically wants to refold them or get rid of them. Okay. If you have more than 10% unfolded proteins, the cell dies. Something like that. Data that I've read. And when there's unfolded proteins, the cell makes a set of proteins called the heat shock proteins. Like chaperones and proteases. Chaperones try to refold, and proteases destroy the unfolded proteins. And these will be our X. Okay, so we have a system where temperature makes unfolded proteins. Are the
unfolded proteins problematic in and of themselves, or just because that they're not acting as they're supposed to?
What's the problem with unfolded proteins? So they're not acting as they're supposed to. That's one problem. You want to maybe refold them. And they're also causing other problems. They stick to things they shouldn't, and they form aggregates. And they cause different kinds of damage. I don't know to list all the kinds of damage that they do. But at least those aspects are bad. And I guess each kind of unfolded protein, can have an unexpected effect because the chemistry inside is specific to that protein and the other proteins that are there. So, yeah.
It's not just that they're useless, they're damaging.
It's not only the damage directly from losing that protein, it's also, you might call collateral damage or glue, like sticking to where you shouldn't and taking up space in these almost indestructible inclusion bodies, you call them. Right. Is anybody here an expert on heat shock? Nice deep sigh of relief. Welcome. Shalom. But this is a universal. I think all organisms, or almost all known organisms, have a heat shock system. It's very important. It's also just used for, even if there's not a big temperature raise, some proteins just need help with the chaperones to refold. It's really has a lot of uses. Now, I want to tell you how the E. coli heat shock system works, a little bit of biological details, like you asked for in the feedback. And it's kind of beautiful. It's kind of a tangent, but we're going to just want to show you this beautiful circuit in E. coli. So you have, what you have is this, basically a transcription factor, a sigma factor. We talked about that yesterday. It's the heat shock sigma factor. And it itself is built to look like an unfolded protein. That's pretty cool. And therefore, there's a heat shock protein that binds it and basically destroys it. So unlike most E. coli proteins that I told you are not actively degraded and are removed basically by dilution, this one has half-life of seconds. It's destroyed really quickly. I'm going to correct seconds. I exaggerated. Something like a minute. So it's made, destroyed, made, destroyed, made, destroyed. That's the normal condition. Okay, and when temperature is high, proteins unfold. So we have our unfolded proteins, and X goes to them. Okay, so there's a lot of unfolded proteins. All the X goes to the unfolded proteins, freeing sigma H shock, which becomes stable, right? It's no longer degraded because there's much less X to bind it. Therefore, its concentration shoots up. So here we have fast response time because of the rapid degradation rate. Once you stabilize it, because it's destroyed so quickly, once you stabilize it, right? This is the fast response time you gain from rapid degradation, and activates the X genes. So this sigma factor goes and binds and activates the genes, the heat shock genes. So that's the circuit. Very elegant. You make sure that the signal is unfolded proteins, exactly the signal you want to remove. Now, once you make more X, suppose you get able to get rid of the unfolded proteins. Why? They fold them or degrade them. What happens to the sigma factor? There's no more unfolded proteins. So the X goes back and bothers and destroys the sigma factor, shutting the system off. So it automatically shuts the system off when the signal goes away. And X is so that's the heat shock system. And that's the prototype for the circuit we're going to, homeostatic circuit we're going to discuss today. Any questions about the heat shock system? In mammalian cells, this works a little bit differently, which is also interesting to think about why, but that's the bacterial one.
Could you expand again the beginning? The sigma factor is always in the cell?
Yeah, the sigma factor is always made and degraded fast, right? It's made a lot and degraded a lot because if there's no unfolded proteins around, the X, the level of X that's in the cell, the cell always has some heat shock protein. It gets rid of it. It's always made. In fact, it's always made. There's a detail where its production rate is also controlled by temperature, but we're not going to deal with that here. But
So sigma is always made and X is always made, and only when they're not binding together, so X is produced more.
yeah, let me draw the dynamics. It will become more clear to you. The point is when there's unfolded proteins, X goes from here to here. Sigma H isn't degraded anymore, and its concentration goes up, making more X. Okay? That makes sense to you? Anybody? I'll draw some graph of the dynamics. Okay, so and the X acts to decrease these unfolded proteins. So that's this circuit here. And now I'm going to write down the equations. So let's spread your space here. Right, so I'm using this to talk about parameter space. So basically, what the dynamics that we're going to see soon, right? Dynamics we're going to see soon, is if this is temperature. So we have a step of temperature. Unfolded proteins are going to do this, and the heat shock system is going to rise. This is the dynamics we're going to do. So when there's heat shock, we'll see unfolded protein rise, give an expression of the heat shock system, which makes unfolded proteins go down. And what we know experimentally is that they go down to the same basal level that they had before. There's like a Y0, a stable. It's not, the cell always has some unfolded proteins. It doesn't get rid of all of them. You can ask why. We'll see soon that you need to really work hard to get rid of all of them too hard. So this property called exact adaptation. Just like we saw in bacterial chemotaxis. Again, I'm running Y is unfolded proteins, and X is the heat shock proteins. So let's write some equations. First thing we'll write is the rate of change of unfolded proteins is going to be determined by the temperature. So there's some rate where temperature makes the unfolded proteins unfold. And they're folded back by the heat shock system. And as always, they have their removal rate, the degradation removal rate. So this is unfolding. This is removal by the heat shock system. This is just the regular removal. But we're going to see that this unfolded proteins happens on a very fast time scale. That's to say, let's say, seconds. Whereas the rise of X here, this thing, takes minutes. So we're going to assume this equation is at steady state for any given amount of proteins X. Compute a steady state, and then let X change, compute a new steady state, let X change. This is what's called quasi-steady state approximation or fast timescale approximation, okay? Because this comes to steady state in seconds. And therefore, what we can write is that because I can bring Y over here, I can write that Y is A over alpha U minus B over alpha X, which means, makes sense. Steady state, if I have more temperature, I have more unfolded proteins. If I have more heat shock proteins, I have less unfolded proteins. Nice deep sigh of relief for us, see? And I'm going to now also use dimensionless variables. Since I'm going to work about parameter space, I don't want these extra numbers here. So I'm going to think about temperature in units of A over alpha. That's to say, this U will be temperature, but in units that give me directly the steady state unfolding protein. So I'm going to, another way of saying that I make a new variable, U, which is the old variable, divided by that parameter times A over alpha. U nu. And X nu is just X times B over alpha. In other words, I'm going to measure the heat shock proteins, in units of their effect on Y. So for example, there's a number here, B, which has to do with the catalytic activity, or how efficient the heat shock proteins are in reducing unfolded proteins. That's a property of the heat shock proteins. What we're going to assume here is natural selection over the billions of years optimize those proteins to be as effective as possible. Why do I do that? Because say if there was a mutation that made them more effective, I can get the same effectiveness with less X proteins, right? So it's there's this very strong and that saves me some costs. So there's a very strong push to make them as effective as possible. So we're going to assume evolution made those heat shock proteins the best machines possible. We still have to make them. So we have one equation. So our system equations, we're going to have one equation, which is this simple equation. And now we need an equation for how x changes. X. So here we're going to use changes according to integral feedback, which we learned about like with the temperature control, if you remember, which is the robust solution to making y have exact adaptation back to its steady state. And that equation is what did I write here? I said that the rate of change of x is proportional to the error, how different the number of unfolded proteins is from this set point. And the way this works is, you can think biochemically, when there's unfolded proteins, just like I described to you, the chaperones bind unfolded proteins, they don't bind the sigma factor goes to the gene makes more x. So when there's unfolded proteins, you get more x. And this term here is the removal of x, and in this model, the removal of x is saturated, it's removed at a constant rate, and if you assume that, you get this kind of equation. This was, by the way, the first people who described the heat shock system this way were Mustafa Hamash and Hannah El Samad, were control theorists. Okay, so we have our two equations. So again, here we used quasi-steady state approximation, very commonly used in biology. Now, why does this give you exact adaptation? You see that when y, so how does this work? How does this work? We start with some temperature here. Right? And after a long time, this term guarantees that y equals y at 0, because the only way to get a steady state is if y equals y0. That's the beauty of this circuit. And I can change any parameters here, any parameters. I'll always get exact adaptation. If I change y0, I change the level, but I always get exact adaptation. There's no way to break exact adaptation in this circuit. So I started some temperature, U0. Y is y0 at steady state. X, because of this equation, x is just y0, is just u0 minus y0. Okay. And then temperature goes up. When temperature goes up, see, x still hasn't changed. X is slow. When temperature goes up, there's unfolded proteins, because the old amount of heat shock proteins can't compensate. And that's why you have this unfolded protein response damage. Y now is bigger than its steady state. Therefore, this term here is positive, and you start producing x. You start producing x, you start producing x, you start producing x. The more x you have, the less y you have, because of this equation. Y goes down, 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 down. When does the process stop? It stops when x stops being produced, when y equals y0. We have exact adaptation. But x is high. If we go from u0 to u1, x is now u1 minus y0. X is the internal variable, the amount of heat shock proteins. If you look inside the cell, you can tell what temperature it adapted to, right? And when you go to 42 degrees, about half of E. coli is heat shock proteins, and then it dies. Can't stand it. And
you have another step.
Okay, if you have another step,
Accumulating more x.
yeah, you keep accumulating more x and more x until, as I said, you reach the upper limit.
What's the limit of
it's around 50% of this biomass. That's at 42 degrees. And then it can't, it presumably can't have enough of the other proteins to live, yeah. Or for some reason like that, yeah. It's like, there's a limit. Of course, there's a limit to, the theoretical limit, 100% of the proteins. For instance, you need to make heat shock proteins, you need ribosomes. You need to make heat shock proteins, you need amino acids, you need RNA, you need, so you can't.
Doesn't the heat shock affect X?
Yeah, it also affects X. Yeah. But let's ignore that here. Yeah, sure. That's another problem, right? Okay. So, in fact, these equations have two parameters. These parameters are k and y0. Because I normalized out all the non-interesting parameters, like with the units of temperature and the catalytic activity of the heat shock proteins there. And because, of course, I want to work with a two-dimensional parameter space, because I want to be able to draw it for you. Right? And those are the parameters that can be evolved and independently. So y0 sets the steady state, the baseline, the steady state damage. What does k do? K sets the speed of the response. So if I increase k, x is made more quickly. How should I do it? I'm going to, this is, if you reduce k, you get a slower response. You get to the same steady state, x and y and everything, but just a slower response. So k controls the speed. Y0 controls the, that's our two parameters. That's what I meant to say, that how parameters affect the dynamics, right? And now we're going to draw our morphospace. Any questions about this? If something's not clear, if I didn't explain myself, this is the time to ask, because other people are. Yeah.
How is X degraded?
According to this model, according to this model, X has a constant degradation rate. It doesn't depend on X itself, constant removal. And that's, of course, can happen if degradation, for instance, if degradation machinery is saturated or something like that. But that's an approximation in this model, and apparently it's a good approximation of the biology, according to Hamash and Samad. Yeah.
Two questions. The first one, could you say again what's the name of protein X?
So X is there's maybe 50 different heat shock proteins, and another approximation that we're doing here is lumping them into one. That's very important to say. Okay. They go up and down together generally. There's also slight temporal order, but that's also less important. Yeah.
Second, can you explain again quickly how you cancel the minus b?
This thing?
Yeah.
Yeah, I
No, I mean,
this
like,
this parameter.
you said that.
This thing?
Maybe I didn't understand you correctly, but you said something about the fact that since it's in a different time scale, so.
Oh. Okay, the quasi-steady state approximation. So we have here a differential equation for how x changes with this k. So it's a much slower rate than how this differential equation changes, because these numbers here, especially these, are very, very fast, because temperature makes proteins unfold in a second, and the heat shock proteins eat up those unfolded proteins also in seconds or minutes or something like that. And here we're talking many minutes. So I can safely assume that if at this moment here, with this level of x, I can compute y, let it reach steady state, and then plug in the value of y here, make x change, and make y reach a new steady state, because this is much faster. So the quasi-steady state assumption is to assume x is a certain number, to make this equal 0. That's because no more changes. And then you get this equation. I moved y here, so y equals u minus x. And I replace the differential equation with an algebraic equation. That makes things easier. I could also have remained with actually the differential equation for the purpose of this discussion, because it also end up to have only these parameters because I normalize out all the other ones. So I just wanted you guys to know about this possibility of making a quasi-steady state assumption. It makes things easier to solve. Did I explain it better? Maybe. Okay. All right, so let's go to parameter space. Our parameter space has k and y0, right? And to proceed, let's look at the biology of the circuit. And I'm going to ask you, why is it that a cell doesn't make a heat shock system that just gets rid of all the unfolded proteins? Make more and more X. It's a rhetorical question because making X proteins, like I said, costs resources from the cell. And therefore you get a sense that the heat shock system, these kind of systems, they must balance at least two tasks. The first task we'll call effectiveness, which means have as little damage, that is Y, as possible. The other force is we'll call economy. Make as little X as possible. So we have two tasks. It's a multi-objective optimization problem. Just like in engineering, or as graduate students, when you think about how you balance your time, or multiple tasks. Therefore, according to what we learned last time, we can think about archetypes, which are circuits optimal for a single task. So, for example, archetype 1, the effectiveness archetype, the most effective system we can think about how would it look like? If you have a step of U, Y would have is almost no change, and also its baseline level would be 0, basically, right? X would go through the roof in order to achieve that. So the optimal, most effective system has y0 equals 0, and k goes to infinity. Yeah.
That's not necessarily true about k. In the most effective system, we have y0 equals 0. And that's it. You don't really care about k if your attempt is to just minimize y0.
Okay, so if y0 is 0, it's fine. But now if you change it, according to this circuit, y0 is 0, y equals 0. But now u changes, and you have some y. So x has to change. So in order for it to quickly get rid of it, I want k to be as large as possible. So, does that make sense? We can't just wave our magic wand and say y equals 0 without a heat shock system. That doesn't work. We still have to be bound by some rules of physics, even though we're talking about theory here. Okay, what does the economy archetype look like? Economy archetype is as if the system didn't care about damage at all. It only cared about saving. So what happens if you're too stingy? x is 0, and y basically tracks u. So there's no, the effectiveness of this archetype is to have no heat shock system. And the way that works is that you put k equals 0. So how can we have no production of x? We put k equals 0, so that this is 0. And there's another way to make this side 0, which is to make y equals y0. So in fact, this is a little bit subtle point here, but since y equals u minus x, according to the equation over there, and we want x equals 0 here, then y equals u, and therefore the steady state is the average u. So the steady state effectively in that circuit is just the average temperature in our units of temperature equals unfolded proteins. So the effectiveness archetype is here. K equals 0, economy. All right? And y0 basically changes with temperature, so y0 is the average unfolded proteins given the temperature. So we have these archetypes. And now, based on what we learned last Thursday, can you erase these equations now? K goes to infinity, but it could be that there's a maximal K. You can't physically make more than, let's say, one heat shock protein per second, one RNA per second, and one protein per second from that RNA. So there's some limit to how much quickly you can make heat shock proteins. So you can put in here a physical limit. We discussed this last time. Or say it goes to infinity. But what we predict in this case is that you get that all systems should lie on the Pareto front that connects the archetypes. Now there's a paper by Pablo Sekeli, who's sitting here, that analyzed this system. Where's Pablo? Over there. Mayo, Sheftel, 2013. Analyze this, and you can solve exactly this problem. You have to assume what the economy performance functions are, etc. And you get kind of a slightly curved Pareto front like this. And the point is that if you have a circuit here with a different K and Y0, there's a circuit here that's better at both economy and effectiveness. And therefore, you can erase this point. That's what's the Pareto front. Right, so yeah, it's on the so what the shape of the Pareto front is, if it's exactly a line or a slightly curved line, depends on the performance function. So this is the economy archetype, and here are contours of economy, and here are contours of effectiveness. And the Pareto front is basically the points where these contours are tangent. So if you assume, for example, that you want the shape of these contours can affect the front. For instance, if you want to minimize, this is just a technical detail. If you want economy, if you interpret economy as minimizing this, and effectiveness is minimizing the damage in this way, then you get a line that looks like this. It's very close to a straight line. But the point is that if I have a point off the line, there exists a point on the front that's better at both tasks. And therefore, this analysis suggests that in this situation, if you look at different damage response systems, by the way, I want to stress that the same model has been used effectively to describe other stress response systems, like DNA damage response repair, when your DNA is damaged and you make proteins that repair. You have an integral feedback circuit. Hormone systems, like if your calcium in your blood drops, like when you, after giving birth, you make milk and it has a lot of calcium in your blood drops, there's a hormone that takes care to release calcium from your internal body stores and bring it back to the basal level. Or glucose homeostasis, where you eat something and you want your blood glucose to go down, et cetera, et cetera. Been described with these integral feedback systems. So this parameter space might capture many different systems. So you can ask, if I look at a heat shock system and a DNA repair system, where do they sit on this curve? Or if I look at the heat shock system from different bacteria, where do they sit on this curve? And Pablo did that for the data available. And for E. coli heat shock system, you get something that's very close to the curve. The K is like 1 over 20 minutes or something like that. So 20 minutes response time. And let's say DNA repair system is here. So they're actually kind of towards the effectiveness archetype.
The points are different systems or different?
Different systems
Bacteria?
in E. coli. Different systems. And there's also another point that comes from a different bacteria. That's when you look at heat shock from a different species. It's called Buchnera. So this is E. coli. E. coli lives, in different temperatures. Outside, it lives in room temperature. In the body, it lives at 37. If you have a fever, it lives at 42, right, or maybe less. Hopefully not 42. But Buchnera. Buchnera is a bacteria that's a cousin of E. coli. But it left the city and went to live in the village inside termites. So when they have a family reunion, Buchnera has to bring the whole termite because it can't survive without it. And the termite gives Buchnera all the amino acids and the sugars. And Buchnera is essential for the termites to mate. So it's really a symbiosis kind of. And this Buchnera is extremely economical. We can see it in all its biology. Instead of 4,500 genes like E. coli, it has only a few hundred. It has only a few hundred genes. Lost almost all its transcription factors, so it almost has no gene regulation. It makes things in a constant rate, at least from the point of view of regulation. And it has no heat shock response, or a very minor one. So when you change temperature, it has heat shock proteins, but they don't change with temperature. So it's here. And it'll be very interesting, once technology is here, or it's already here, to put more systems and see. See, it's easy to falsify this theory if you have points here or here. Then you think maybe there's another task or maybe there's something wrong here. So to summarize this part before we take a break, what I'm inviting you to think about now is circuits on a new level of parameter space, with the potential that there's simplicity not only in the number of circuits we have, but when we take a circuit and focus on the parameters, potentially there could be a low-dimensional situation with its parameters, especially if there's not many tasks that are crucial. And to help us think about that. So we're going to take a 10-minute break. Wanting to know, is there any questions, burning questions about circuits, parameter space, the homeostasis, integral feedback, what we studied last time, the idea that circuits might fill out a low-dimensional manifold in parameter space.
The K and the Y0
where do they come from in the biological circuit?
The role of K and Y0 in the biological circuit. So this is X production. This is X removal. So the K, you might say, is the rate of X production per unit Y, and then KY0 together. So you can get Y0 by taking the removal rate, dividing it by K, you might say. Okay. And usually we learned that you have an X here because removal is proportional to X. Like this is, but in this model, it's like there's some machine that gets rid of X and it's saturated, something like that. So that removal is the first. It's what's called a zero order removal. It doesn't depend on X. It's not a first order removal. And why that is something we need to look at the biochemistry. Why? I actually prefer this fold change detection variant of this circuit, where the difference is I put X here. And this makes things much more natural. Now what happens is that X is removed as usual, and the production is an interaction between X and Y. And that's like in the real system, you have those chaperones need to interact with unfolded proteins in order to get away from the sigma factor. And so this is a more natural equation. It still has integral feedback because Y minus Y0, the only steady state is Y0, but it has this X term. This X term actually gives fold change detection potentially. So it's a nicer model. So what I wrote here is the model that we worked on in this paper, and it comes from those earlier works in the heat shock system. But I think you can make a more accurate model and you get the same properties. Yeah. So when I write my book chapter, maybe I'll use this model. It's in the new second edition. So thanks for that question. Any other questions?
Why heat shock? And what if the heat rises gradually and not in a step function?
Oh, so why is the system called the heat shock system? I guess it's historical. It's the way people did the experiments, as far as I know, is take bacteria growing at 37 and put them in a 42-degree water bath for, let's say, a minute. And then counted how many bacteria survive. Right? And looked for mutants where bacteria survive less. And those mutants were proteins and they call them heat shock proteins because it's a mutant where E. coli doesn't survive a heat shock treatment, which is raising the temperature like that. So it's a historical name. But in mammals now they call it the unfolded protein response, UPR, more accurate. Because also, by the way, ethanol, when you drink alcohol, because of what alcohol does to the hydrogen bonds in water, it also causes unfolded proteins. You can get unfolded protein response also with ethanol, for example. Did I answer your question?
No, the question is if you rise the temperature not in a step but gradually throughout a long period of time.
If you make, let's say, a ramp of temperature. So this is a very interesting point. Integral feedback, linear integral feedback like this, cannot lead to exact adaptation for ramps. You need a double integral for that. So if you do this experiment and ask whether you get exact adaptation, compare a step to a ramp, you can actually figure out if it's linear integral feedback or exclude linear integral feedback. And I don't know the answer to this question. I mean, I know in bacterial chemotaxis, if you do an exponential ramp, the system doesn't have exact adaptation. It kind of goes to, it's like climbing a gradient. It always has lower adaptation. That's one piece of evidence for the kind of models we have now. But maybe more simply to answer, you can take your computer, take these equations, and with MATLAB, see what happens. But those kind of biophysical experiments are very informative about ruling out certain mechanisms. All right. I'm going to suggest now that we play a little game together that has to do with optimality in metabolism. And I'm going to write down the rules of the game. But each of you should have a piece of paper to write and a pen. So, Jean, maybe you could look and see. If you don't have a piece of paper and a pen, you can raise your hand. We have prepared writing materials for you.
Only before you raise the last part.
Yes?
Quick question. If you have a higher level of HF proteins now, shouldn't your Y0 be lower than the original? Like, shouldn't it go? If you have a higher level now of HF
Here,
proteins,
this state?
yeah, so shouldn't the Y be under Y0 now?
So the way these equations work is to magically y equals u minus x. So I change temperature. I have the old level of x, so y goes up. X starts working. Y is higher than y0. X is made. The more x is made, the less y there is. When does this process stop? When the x dt equals 0. No more rate of change of x. Where does that happen? Only when y equals y0. So this guarantees that you return to the same level of y0.
I know it does in the equation, but I mean in real life, like you have more HF proteins than, at least to me, it would seem that you have a lower level of unfolded proteins.
If I now go into the cell or express from a plasmid some heat shock proteins, let's see what happens. I express some heat shock proteins from a plasmid. Y goes below. But because of this equation, it's actually heat shock proteins have a negative production rate or bigger removal. And the system also adapts back. So that's also a prediction here. If you have too many heat shock proteins, cell will get rid of them. Or as it's growing, it will make less of them, so the dilution eventually will get back to the level that you're supposed to have. Here's an experiment to do, right? When you have a model, you also have new experiments to do. That's the nice thing. And then you revise your model. Okay, so I'm going to ask you to play the following game. It's called the game of the pentose phosphate pathway. And the idea is it has to do with carbon-like molecules, but we're going to play it as if you have bags with stones in them. So you have, the game will be to take 6 bags with 5 stones, that's a total of 30 stones, and end up with 5 bags of 6 stones, also 30. Right? So we're not going to lose any stones along the way. And the rules are, in each move, you take one bag, you take some stones and move it to another bag. That's the rule. That's the moves. Okay. In each move, you can transfer either 2 or 3 stones. And 2, you're not allowed to have 1 or 2 stones in a bag. Okay, so those are the moves. And your goal is find the minimal number of moves to go from 6 bags of 5 to 5 bags of 6. Any questions about the rules? I'll just give an example. The only thing you can do actually in the first move is to take from one bag 2 and move it to another bag. So 5 and 5 become 3 and 7. Why can't I move 3? Because then that 5 will become a 2. That's not allowed. Okay? So please enjoy. We're going to take 3 minutes to write this down and yeah,
The remaining move them.
yeah, you can take the 3 and move them to another bag. Sure. You're allowed to have 0. Good question. Okay. The idea here is to try. Okay, you don't have to solve it. It's to try. So I want everyone to try. And you can also consult with the person sitting next to you. That's okay. That's fair. Or maybe behind you. So I'm wondering who's going to be the first to offer a solution here. You have a solution? Great. So we'll wait for another minute, okay? And
You're taking 5, you're moving 2.
okay, I'll let you draw it out and explain, okay? So it looks okay to me. All right. Yeah. Yeah, that's right. Count the steps. See how many steps you did. Nine steps. We have one solution here with 9 steps. Does anybody have a better one? 8 steps? Anyone have a better one? Huh? 5 steps, that's too little. That's not possible. 7 steps? What? You can't move 1, correct. Yeah.
How do you count steps?
How many times you do this? Look. So you have 7 steps? 9 steps? 7? Okay. All right. So can I ask you with 7 steps to come up? Would you be willing to? Oh, check. It's okay. By the way, this is not the point. Is to be a fast problem solver. It's not the point. I just want you to experience. Yeah. Okay. Something that metabolism has to solve too, and to see. Okay. So what's your name?
Gal.
Gal, and what group are you in?
Avi Ben Zev, cell biology.
Avi Ben Zev, cell biology. Okay. So first, let's give him a nice round of applause. Whatever the solution, if it's right or wrong, you get an applause also.
It, yeah.
Okay, you can handle it. Good.
Okay, so the fastest way to make this 6, taking 2 out of here, 3 and 9, and then take 3 back.
Wait, hold on, slow down.
Taking the 7 and the 5 to make this 6 as fast as possible.
Okay, hold on. So you take the 7 and the 5 now, and what do you do? So we take a 7 and a 5.
Taking
7,
2 out.
2 of here.
Okay,
so you get a 3 and a 9. Okay.
Then switch between them again. This time I'm taking
3
from
okay.
here back,
Okay.
and 6 and 6.
Okay.
Now I have to take this.
This 3.
Yeah, because
Okay.
there's only one option to do with this.
Okay.
So I put it here.
So it's a 3 and a 5.
It's 8 and 0.
Okay. So let's say that you did this, right? 3 and 5, 8 and 0. Yeah.
And to make this as fast as I can, 6 is just taking out 2.
6 and 7.
And then 7 and 5 is exactly like here.
So can you draw that?
Mm.
7 and 5.
2 steps, it's 9 and 3.
Okay.
And
6 and 6. Okay, so let's count the steps. We have 1, 2, 3, 4, 5. 1. How many steps?
I have
1,
7 here.
2,
3.
3, 4, 5, 6, 7. Is that right? Okay, let's give him a round of applause. That's not the solution that I know, and I wonder if it's. Does anybody have another solution? 7?
I have one, but it's really, no, it's 9, but it's
9. Yeah, there's another 7. You have a 7 that's different. Is this a joint effort?
Yes.
And what's your names? Chen and Liran. What labs? What groups?
I'm from computer science, and
Okay.
Liran is doing rotations in bioinformatics.
Okay, great. Which lab are you in now? Monipaz. Okay. Do you have a mentor already? In computer science?
Not yet.
Okay.
It just started in March.
All right. Well, let's put it here. Second solution. Okay. It's pretty amazing, no? There's another solution. Okay.
Okay, so we started the same way.
3 and 7. Yeah.
And then 5.
Okay.
After that, we do 6 and 4. We move 3 from here to here.
Okay. Okay.
And then we take this,
Okay.
and we make it 6 and 3.
Okay.
After that, we take this and do it again.
Yeah. So it's symmetric, right?
Yeah.
So after you do these 3 steps here, you get 6, 6, 3, right? 3
Yeah.
steps later,
And then you just
take
combine these.
3 and 3 to make a 6. Okay, so it's 3 plus 3 plus 1, 7, and symmetric, right? Because you can work just on these 3. Okay, big round of applause. Very cool. So 2 different solutions. Let's just say the maximum number here is 7. There, the maximum number is 9. Okay. And now I want to give you the context why I asked you to do this game. So it's called the pentose phosphate pathway game, because this problem, you can think about a certain part of the metabolism of E. coli as having to solve this problem. And I don't want to erase this beautiful solution, so I'm going to write it. So in the pentose phosphate pathway, E. coli takes 6 5-carbon sugars and converts them into 5 6-carbon sugars. These are called pentoses, and these are called hexoses. And the reason E. coli needs to do this is because a lot of sugars in the environment, like arabinose, are 5-carbon sugars, and it basically builds an entire biomass from glycolysis, which is 6-carbon sugars, which it splits into 2 3s, etc. So also in the middle, it can use this to generate 5-carbon sugars running the pathway in reverse, which are important for DNA, the ribose backbones of DNA and RNA. So this is an essential part of metabolism. So this game by Melendez-Hevia, who was a scientist in the Canary Islands off the coast of Africa, abstracted this part of metabolism and said, let's think of, just count the number of carbons. So we have 6, that's the stones, right, the carbons. And look at the enzymes here. And the enzymes, you can do this. These are called ketotransferases and aldolases. The enzymes transfer 2 or 3 carbons between these carbohydrate molecules. And you're not allowed to have a single carbon, which is CO2, or 2 carbons, which is acetate, because those go out of E. coli. So let's say those are the constraints. And the minimal solution, this one, is what's found in the actual pentose phosphate pathway. The symmetric solution with going up to 7 carbon, this septoheptulose, it's called, in this configuration. So the minimal solution, you can prove that the minimal solution has 7 steps and appears in the real pentose phosphate pathway. And we see here that there's another solution of equal length. There's no solutions that you can prove, no solutions of less than 7 because of the number of things you need to do here. Basically, you can show that. I don't want to do it, but you can also just enumerate all the steps. And this kind of finding, so is a kind of finding that's very exciting for people like me, come from physics, computer science. Very annoying sometimes for biochemists because it abstracts the very complicated biochemistry and molecules into something very simple. Because molecules aren't just the number of carbons in them, and enzymes aren't just can do anything like transfer from any molecule to any molecule. It really depends on exactly the details and the energies, etc. So. But still, there's this elegance, and I wanted to show it to you and to tell you a little bit about developments after this, more recent developments of this idea. The idea of metabolism as a game, kind of a minimal pathway game. Yeah.
But you might also want not only to minimize the number of steps, but also minimize the number of substrates because the enzyme might not be able to deal with a wide range of substrates.
Okay.
And then the right solution on the right is more optimal.
Okay, good. So maybe you can say, how can we now tell the difference between these two solutions? And we can ask, first of all, you can ask why does it make sense or to have a minimal number of steps at all? And also maybe there's multiple tasks, right? For example, these intermediates, these molecules, the 3, 7, 6, 4, in the middle, sometimes are just there to convey mass, you can say, from the beginning to the end of this pathway. But there are also, it's not good to have too many different kinds of these intermediates because they have their own chemistry and they can be toxic or they can accumulate. So maybe you want to minimize the number of substrates possible. I mean, based on what we talked so far in the course, you can understand why minimal number of steps might make sense because each step like this, you need to make a special enzyme. You need to make several thousands or tens of thousands of copies of a protein. And that, again, costs resources. So the less steps you have, you can say that translates or relates to needing to make less machinery to carry out. But of course, it could be that you can have, let's say, a game with more steps, but it so happens that the exact chemistry is so efficient that you need very few enzymes. So it's possible not to have a minimal game in principle, right? But so this kind of argument is not as simple as I'm telling you, but still it's pretty exciting for me that a game like that it takes us some minutes to solve, and it's not trivial, and it matches what evolution kind of found. Yeah, there's a question. Sheila, nice deep sigh of relief. You can fill them in later on what they missed. Yeah.
You mentioned that you need a different enzyme for each one of the steps, but if you're looking at a symmetric solution, so you need less enzymes than non-symmetric.
Good point. So it could be, if we count not just the number of steps, but the number of kinds of steps. For instance, this 2 5s, and what we need to do here, the symmetric solution, it's the same enzyme. It's the same enzyme. Whereas here, I don't know. I don't know. I mean, of course, this step is the same, right? I don't know. We need to study this, see if it makes sense. But I wasn't expecting this. I want to say. Thank you for. It's nice for me as a teacher to have the first time I'm trying to play this game in class. So, maybe there's more kinds of enzymes need to be made here. Correct. I just want to say one caveat is, it's.
One more.
There's one more.
One more enzyme.
Oh, thanks. You counted it. Okay. Right. So I think here there's 3 kinds of enzymes, and here. 5 and 4. Okay. There's really this transketolase and transaldolase, and this one is called it's something that splits the 6 into 2 3s, like in glycolysis, working in reverse. On the other hand, if you have more flux that needs to go through an enzyme, you also need to make more of it. So it's not. This is also to take into account, right? But it's a very good thinking what you guys are saying, to start thinking of multiple tasks in metabolism, which is also an open field. Usually, people, because of the dogma and evolutionary thinking of a fitness function, which is almost like saying one task, people haven't been thinking about multiple tasks. There's examples all throughout, but the sporadic papers in the last 30 years or something, but it's not certainly not in the dogma. But if you start thinking about multiple objectives, maybe we can in the end try to understand what is the constraints for metabolism. But of course, there's also energy constraints. I want to get a little bit deeper into it, not too much, but more questions. I see this is stimulating. Yeah.
And also over there, since you can separate into two smaller tasks in the beginning, then you can do it in parallel.
Okay, so this, as we said, is symmetric. And if I understand what you're saying is, we separate it into two tasks. The tasks are to take 3 5s and turn them into 6 6 3, right?
Yeah, you can do that in parallel.
You do that in parallel, and then you join the 2 3s at the end.
So that can take less time than doing something else.
Less time, in the sense that you're less dependent on waiting for previous reactions to work. So I would say at steady state, there's no sense of before or after. But if you now want to start up the system, then if you have more steps, maybe you can calculate that. I don't know if you're fast. How many? What's the response time? So if each step takes one unit, what's the response time of this one and that one?
But you can do this one symmetrically as well.
You can?
Yeah, because after you turn the first step 3 5 to 6 6 3, right?
Okay, so you're saying turn the first.
And then it becomes almost the same as the symmetric solution, just with.
Okay, hold on. So let's do it. I mean, why not? We're here to enjoy ourselves, right? The idea is to enjoy ourselves in science. And if we're playing games, why not enjoy ourselves? So you say 5 5 5 goes to
3 and 7. Actually, and then the 5 and the 7 to 9 and 3. And then the 9 and 3 to 6 and 6. You move 3 from
6 and 3, basically, right?
No, 6 and 6.
6 6 3, and we do it again, and then we connect these, right? Correct? Okay, so they're both actually symmetric solutions. Is that correct?
But here you have a 9, which I guess.
Okay, so a 9. Okay, so then you can ask, is there any cost or any difference between having a 7 and a 9? So the body ourselves do have these long carbon chains, fatty acids, for example, and so, but typically not. I don't know of sugars that are 9. So, good question. I mean, it's pretty impressive, you guys. So this generated some mild discussion, mostly forgotten because until recently, where more physicists and computer scientists entered biology, got excited about this kind of simplification. And I want to tell you about one more recent work that tries to use this idea for more complicated metabolism. I just want to say the problem of metabolism, you probably know this, is extremely salient now because it's the key, for example, to being able to make, let's say, biofuels out of organic material. If you can make E. coli or other organisms make ethanol or make diesel, biodiesel. So it's all how to make E. coli, take the carbons and do something different with them. Or to make more plants have more food yield, etc. So there's a lot of interest now in understanding metabolism and, of course, basic research questions. So, maybe you can prove are minimal solutions always symmetric in this situation? That would say something about metabolism. So I want to tell you about a work that tries to look at a bigger chunk of metabolism. So we're going to examine the idea of minimal walks in a larger chunk of central carbon metabolism. So I'm going to try to sketch your central carbon metabolism. This is conserved very, very strongly from E. coli to human beings. All the enzymes, yeast, worms, flies. It's like major. Every biologist knows what I'm going to say. Should know. There's pathways. I'm going to just draw sticks like this. There's a part called glycolysis that takes 6 sugars, splits them into 2 3s, does some more stuff with them, puts them into the TCA cycle that generates a lot of ATP. I'm not drawing everything here because the details aren't very important. And here is the pentose phosphate pathway. And here is the external sugars, like arabinose gets transported into pentose phosphate pathway here. Glucose goes here. So different sugars converge on this. And this makes ATP and biomass, basically. This is, again, when you study biology, you learn the 10 enzymes of glycolysis, and you learn what they do, and then you learn TCA cycle. And this work is by Elad Nur, Ron Milo, and the idea was to add also a little bit. So, okay. So if you just try to do this, the pentose phosphate game, if you just try to play the same simple game, where each molecule is represented by the number of carbons, you find plenty of short circuits. What does that mean? Smaller paths. Shorter paths than what's observed. So you say you can have a minimal path, let's say, that goes, instead of all these steps, you go from here to here in a couple of steps or something. So it doesn't work. More constraints. And so to approach this, we want to still play a game, but make it more biologically, chemically, let's see. So the approach was like this. Take all known kinds of enzymes that work on carbohydrates. So here, in this central metabolism, there's no nitrogen and no sulfur. So it's, again, a simplification. Just carbon, phosphorus, oxygen, hydrogen. And it turns out there's something called enzyme classification, EC 1.1.3. Those of biologists know this. So it turns out there's only 30 kinds of reactions. For example, taking something with 2 carbons connected to something and passing those 2 carbons to something else. And that's just like the step that you did with the 2 stones. That's one of the 30 kinds of reactions. And the second simplification is to consider each molecule as a string. I say string like in computer science. So you say, right, CCH, CCOH, CC, right? And describe each of those enzyme types as an operation, like in computer science, on the string. So basically, if you have, and you work this enzyme, you go to, right? You just run the string operation. Wherever you can find something in the string that the enzyme can work on, you do that operation. Now, real chemistry, this contains many more reactions than are really possible, right? Because of energy constraints like delta G constraints and all kind of steric constraints. So if this is all the potential reactions, and this is the real possible reactions, you're considering a larger space. But if you find a minimal solution here, it's guaranteed that you can't find a real solution that's more minimal. So this is kind of a... If you find that the minimal solution is 7 steps in this big space, there's no way to make it in less than 7 steps in this smaller space. So that's... But this gives you an algorithm now. If I give you some molecules in the beginning and some molecules in the end, this can find the minimal path. How does that work? You take your beginning molecule. This is your end molecule. It's actually a set of molecules here and a set of, let's say, your 6 pentoses and 5 hexoses, right? Like we did. And now you work all possible steps with all those 30 enzymes. You do all possibilities. And then for each one of these, you do all possibilities. On average, a typical biochemical metabolic molecule, there's 20 steps you can do. And for each of these 20, another 20 steps. So this grows really exponentially. But you want to find this molecule. And there's a computer science trick called breadth first search, where you do it in parallel. You do all the steps on this molecule, then all the steps on each of the steps, until you find a path. That makes a square root on the number of computations you need to make. So you get a set of molecules, beginning and end, and this prints out for you all the minimal. So you say, okay, we can do this in 5 steps, and here are all the ways you can do it in 5 steps. So if you want to produce, synthesize something, like some exciting new plastic for your iPhone cover, starting from pyruvate, glucose, and a little bit of, I don't know, whatever you put in there, you can find the minimal number of steps. You can do it in 27 steps with these enzymes. And it doesn't mean you can actually do it, but you can't do it in a shorter way with real enzymes. And then so any questions about this software?
How do you know what you can actually do?
So we don't know what you can actually do. We don't know what you can actually do. There's, of course, all the real enzymes. So there's maybe a catalog of several thousand real enzymes found from all different organisms. But there's many more. Because there's so many species. This is also important to know. There's so many species of bacteria. Not to mention, the hundreds of thousands of species of beetles, for example. But species of bacteria, which are, it's even difficult to find species because the genomes are so plastic. The bank of enzymes in the biosphere, we know really a tiny, tiny fraction of it. We can use the software to try to understand if a reaction is biochemically feasible. And there's advances in that to calculate. For instance, you can calculate the delta G between the reactants, and you want a gradient of free energy, for example. But enzymes catalysis can do, like burn ATP and do all kinds of things. you might do steric, so you can't make two carbon atoms overlap physically, right? So you might worry about that and all other chemical rules like this. But there's still not, as far as I know, not a good prediction of what actually works in the cell. But I could be wrong, but but so in this approximation, you take all potential reactions, which is we're pretty sure based on these thousands of enzymes, just 30 kinds of things that enzymes do. Maybe there's another thing we haven't discovered, but that comes up again and again, like the ketolase, allylase, or protein kinase that adds a phosphoryl group to somewhere, or phosphatase that takes away phosphatase. There's 30 kinds of reactions. Actually, usually when you ask a question, I don't understand your question. I always ask you, did I understand your question? And then you say something else. What is it?
The question again was, how can you know which kind of procedures are allowed? Because the way that you made the big group is by taking all the enzymes in all the bacteria
Right.
that you know that are allowed.
Yeah.
So the question is, the path allowed?
No. Okay. I took all the kinds of reactions, so it's not each enzyme is specific. That means each enzyme works on a specific molecule. But what I did is abstract the kinds of things enzymes do. So, for example, take two carbons from something and move it to something else. But it's not guaranteed that that particular something can really biochemically be done. So there's a difference between kinds of reactions and actual reactions. There's 30 classes of reactions. This is one of them. And there's thousands of examples of this kind, each time with a different R and R prime. Okay, it's very good that you insist on understanding your question, because again, you have to realize that you should realize that when you answer somebody's question, there's a big chance that you didn't really understand the question, especially when you're a speaker. So that's why it's good to see if you really understood. In the end, very difficult for a human being to understand another human being. That's the way it is. But we can do it. Okay, so what did the lad and Ron found when you ran that algorithm and saying these are the inputs. Calculate now the minimal paths. Again, they found that there's plenty of shortcuts. You can jump from here to here. You can make a lot of many more minimal paths. But there was also a principle that came around. And that is, I'm going to write down what the principle is. Central carbon metabolism is a minimal walk between biomass precursors. So what do I mean? In this metabolic system, there are 12 special places where carbon goes out to make amino acids, fatty molecules, amino acids, nucleotides. In other words, there's 12 famous molecules like pyruvate, alpha-ketoglutarate, etc. Biologists know that are the basis for making all the rest of the stuff the cell needs: the proteins, the membrane, the DNA, the RNA. And what they found was that if you take two consecutive precursors, the path between them two is the minimal. So the path between any two consecutive precursors, like this one here, 1, 2, 3, 4, 5, is the minimal. All right? One moment. Path between any two consecutive precursors, which are points at which carbon leaves to make amino acids, membrane, etc., DNA, etc., is minimal. Sometimes there's many, like we saw in the pentose phosphate game, there's many possibilities of the same length, and one is chosen. And then you can ask why that one and not the other ones, but it's always the minimal path. So it's like a multi-objective optimization problem again. You want to minimize the number of steps, but you also have to go through, like a traveling salesman problem. You have to go through 12 points. Right? And one possible use for this kind of thinking is, now suppose you take a new metabolic pathway in a new plant, let's say, you discovered a lot in plants, let's say there's, out of this central carbon metabolism, there are these very elaborate biosynthesis pathways that in the end produce something, let's say, that smells like pizza, let's say, right? Timol, right? Or some plant stuff, right? They're so good at making these amazing plant products. So secondary metabolism. So suppose you run your algorithm and you say, look, you can do this in a shorter way. But this looks like it goes in the shortest path to this chemical and then in the shortest path to the end. Then you can say, oh, that chemical maybe has another task. Maybe it's a branch point. Maybe it's essential. It's like if I want to go from Rehovot to Jerusalem, but you look at my my taxi receipts I give to get refunds, and you say I went through Haifa, which is 150 kilometers north and then south for those of you who aren't Israeli and watching me. You say, maybe I had something important to do in Haifa, right? This kind of idea.